you want me to go ahead and put this up?
All right, thanks, Todd. So in the spirit of the panel members not being compensated, I'd like to suggest that the first person whose cell phone goes off during these sessions has to run down to Pilates and buy lattes for all of the panel members. So this morning we're moving over to the Cook Inlet side and examining some of the baseline studies from the Cook Inlet marine ecosystems. That's the last session for these panel members. They'll give some recommendations at noon, and then we'll excuse them and bring on a different panel for the next session on social and cultural resources. But we're going to start off today with a presentation from Dr. John Houghton. Dr. Houghton received his bachelor's from Harvard, he says a very long time ago. He got his PhD in intertidal ecology from the University of Washington School of Fisheries. John is a senior biologist with more than 40 years of research and consulting experience in Alaska and the Pacific Northwest. In Alaska, he's worked in both coastal and riverine environments, all the way from misty fjords to the Aleutians to the Beaufort Sea coast. So this experience has included baseline studies in remote areas, ecological risk assessment, permitting, habitat restoration, NEPA, and ESA response planning, and it includes substantial field research in freshwater, estuarine, and marine environments of Alaska. He has special expertise in the littoral and benthic ecology of southeast and south central Alaska. He is an expert in the ecology of anadromous salmonids and in the effects of perturbations, especially habitat alterations on cold water fish populations. And he's going to talk to us about some of the Cook Inlet baseline studies.
Okay, thank you, and good morning, everybody. As indicated, this is our subject for the morning, as the work that we've been doing in the marine environment, where Pebble is contemplating the potential for putting in port site facilities necessary to support the mine, both logistically and sending equipment up to the mine, but also in extracting or shipping the products of the mining process. I'd like to recognize my co-investigators here, Jason Stutes, Jim Starks from Pentec Environmental, and Hart Krauser, and also several others, including notably Dennis Lees and Sandra Lindstrom. Both of them have worked in Alaska at least as long as I have. So our general study goals are to build upon historic information and update that information regarding this particular part of Cook Inlet. We want to gain a detailed understanding of the marine environment in what I call the IIE, which is the Iniskin-Iliamna Estuary, and we'll get to a map shortly here that shows exactly where that is, basically on the southwest side of Cook Inlet. Our focus has been on fish, benthos, and demersal. That means near bottom assemblages. But we also have been looking at oceanography, water quality, and chemistry, which I'll start out with. Our study goals in that piece of our work are to describe existing conditions oceanographically and the condition of water in the area, and develop a baseline for long-term monitoring with data on water quality, sediment chemistry, and animal and plant tissue chemistry. Here our objectives are to describe the physical oceanography based on existing information. We also have been collecting field data as we sample, particularly as we sample fish populations, and invertebrate populations, we take concurrent water quality measurements during those activities. Physical oceanography, here we are with the map. Hopefully we can see that Anchorage, of course, is, this is exactly what they warned me about. Anchorage is way up here in the upper right side of the map. Homer down here in the central right side in Kachemak Bay. And directly west from Homer, just north of Augustine Island, is Inniskin Bay and Iliamna Bay. These areas get substantial influence of water from the Gulf of Alaska. This map of the general circulation pattern in Cook Inlet was worked out by Dave Burbank back in the mid-70s and hasn't really been improved upon very much with subsequent work. But essentially you see major masses of water entering Cook Inlet from the Gulf of Alaska on the lower right, moving up into the inlet, into Kachemak Bay, north up, generally north up the east side of the inlet, and then a net southerly flow down the west side of the inlet. And again, our study area here is over on the center left side of this diagram. Zooming in a little bit on that, the data were fairly vague about what happens to water in the immediate vicinity of these bays. We do know that there's very strong tidal circulation, so you have a very robust tidal currents generated by the tides that can fluctuate over 20 feet in an extreme tide event. One factor that stands out in this part of the world is ice. Here we see some ice forming in outer Inniskin Bay in November. The influence of ice is still present as late as April in the subsequent slide. We can track the ice in the study area and elsewhere in Cook Inlet through the National Ice Center online maps of ice thickness and coverage, and this is extremely useful in comparing year to year. You can see in the early part of this record here, this graph shows the maximum ice extent throughout the course of each year. Sometimes that maximum occurs as early as January or as late as toward the end of February. And again, we have the little arrow there that shows you where the IIE, or the Inniskin-Iliamna estuary, is located. Water quality in this area is generally very good. Water is much clearer than it is up in this part of the northern Cook Inlet. There are no local contaminant sources, so the baseline chemistry is very good. The water clarity is a little bit murky for diving, however. It complicates how well divers can see and work on the bottom. We've been sampling water chemistry for detailed trace element analysis, both at the near surface, near the bottom, at 3 primary locations. And we have also looked at tidal influences. We typically sample near the high tide, within 2 hours of high tide. But we've also done samples on subsequent high and low tides to see if there's a significant tidal influence. Our primary sample locations, if I can get the arrow going here, there we go. MOPP1 is down at the entrance to this study area, and right there. MPS1 is another key site that's up here and near the entrance to Inniskin Bay, and then MPS4 over to the left near the entrance to Iliamna Bay. So those have been our key stations for doing the detailed water chemistry. All of the other little dots and notations on this chart are locations of other types of sampling where we've done concurrent water sampling for basically temperature, salinity, and turbidity. Taking a look quickly at a little bit of the data. This is mean salinity by month from March through November. Typical pattern here is that salinity is very high during the winter when their freshwater sources are pretty well frozen. It dips down with runoff during the springtime, generally reaching a minimum in late summer. And the interesting thing about that is that this pattern indicates that the water in the study area is actually more influenced by the large freshwater sources in the upper inlet, like the Susitna River and other major rivers up north, where those rivers depress the entire salinity of Cook Inlet when they're running full in midsummer. And the local runoff in the study area, much smaller drainages, much smaller streams, peaks in May and June. So we can see that the influence from the upper inlet is strong throughout the summer. And the same thing occurs with turbidity. As far as water chemistry, this is probably a pretty confusing chart for most folks. But basically, on the right, you see the list of trace elements that we've measured in the water. What this chart depicts is what we call a mean adjusted concentration, where these various metals and trace elements are present in extremely low concentrations. And so if you just look at the raw numbers, they're really all over the map. So what we did here to look at a seasonal signal, where we've done May, July, and September sampling, was take the average of those 3 months for each of these elements and normalize it to the mean. In other words, this shows you how that particular element varied through the year. And what's evident here is that between July and September, there's really not much variation. There's some indication that in our spring sampling in May, some of these constituents tended to be a bit higher in May, and that's perhaps a result of the spring runoff from the adjacent drainages that brings materials that have weathered through the winter down into the marine environment. But basically, the water quality is very clean. These concentrations are all very low. So what we have is a baseline, and that's really what we're after. Turning now to sediment chemistry, our key stations here, again, for the subtidal chemistry are the same as for the water chemistry, with MOPP1 down here in the outer entrance to the study area, MPS1 up in the entrance to Inniskin Bay, MPS4 over in the entrance to Iliamna Bay. And the yellow dots on this chart are where we have intertidal sediment samples. Those we often will take both at the upper intertidal and lower intertidal. And the way we do that for the subtidal samples, we use what's called a Van Veen grab that actually just grabs a chunk of bottom, and we scoop the surface material out of that grab sampler and put it into sample jars using very clean techniques to avoid contamination. We typically sample the top 4 centimeters or a little less than 2 inches because that's where immediate metal contamination or inputs from surrounding sources would occur. We do sample below 10 centimeters where we have a deep enough sample to kind of look at the historic concentrations and see if there's any indication of change over time. And then we have been doing 5 replicates per station. Looking at the data, like the water quality data, the numbers are generally very low and representative of what we consider to be background in the study area. What these graphs show are, 6 different metals that are, and the scale of the bar is shown over here in the legend. But basically these are in parts per billion, and we're looking at zinc is on the right-hand side of each of these little bar graphs. Zinc shows to be the most abundant of those metals, where we have 2 sets of graphs for a given intertidal station. The upper one is middle intertidal elevation, and the lower one is lower elevation. Looking at subtidal, again, we see the same kind of pattern. These subtidal stations aren't particularly deep. It's really only, many of them are 20 to 30 feet deep. The deepest is maybe 50 feet deep at MPS 1 up here and MOPP 1. For another group of elements are present in much lower concentrations, so we separated them out for graphic purposes. These are things like antimony, cadmium, mercury, selenium, and silver. And of these, selenium tends to be the most abundant, but as mentioned, they're really very comparable to levels that have been reported elsewhere around the state of Alaska. Zinc, copper, and chromium concentrations are somewhat higher than other reported data from the general area. Mercury, cadmium, silver, and antimony are relatively low. Arsenic, copper, nickel, and zinc actually do exceed the squirt values, which is a compilation of test concentrations that exhibit some form of effect on biota, on plants or animals. So these 4 elements in the natural baseline condition actually exceed those criteria, but again, that's not unusual in any natural location to have a certain suite of elements that are characteristic of the local geology that do exceed those criteria. Turning to organics now, total petroleum hydrocarbons. This just shows the very gross numbers between MPS 4, the offshore station, and MPS 1. And what this shows is that there are higher concentrations in the inshore stations, MPS 4 and MPS 1. Again, this is totally natural, not unexpected. These stations are closer to sources of plant material that show up in this type of analysis. This is one of those graphs that I think we were warned about not to include, but it's hard to talk about results much without doing this. What this is a look at the relative abundance of straight chain hydrocarbons, very simple hydrocarbons. And what's of interest here is that in this range out here between C26 to C30 means there's 26 carbon atoms in a line. You all remember this from organic chemistry, I'm sure. But between the C26 and C30, the concentration of the odd-numbered chains is greater than that of the even-numbered chains, and that's indicative of a biogenic source or local terrestrial plant matter has contributed. We see the same picture in the subtitle, chemistry. And let's see. Then if we turn to polynuclear aromatic hydrocarbons, these are things that basically run our car oil plants, crude oil, diesel oil, all of them have characteristic signatures of these PAHs, as it's called. And what's relevant here is that in both the intertidal and subtidal PAH, this is called a fingerprint, and this is a way you can identify specific sources of these PAHs. Two things that stand out in these graphs. One is the very high concentration of parylene over here on the far right, and then and that's considered to be indicative of biogenic sources, or perhaps a pyrogenic source, which is the combustion products of burning diesel oil, for example. Then the other thing that shows here is that we generally have comparable concentrations of, there's no simple way to say this, the alkyl substituted homologs of each family of hydrocarbons, and the pattern essentially, again, doesn't indicate that this comes from a local source of oil particularly. So basically what we see in all of these analyses are that we have a pretty pristine environment, and we don't see any indication of local or strong local sources either from coal. Coal can also show up in these types of analyses, from crude oil, diesel oil, or pyrogenic or combustion products. So really we're dealing with a very pristine environment, which doesn't surprise any of us, I don't think. Looking at tissue chemistry, the reason we do this is to try to get a baseline again of the levels of some of these potential contaminants in the tissues of plants and animals that live in the study area. So we've sampled a wide variety of species, plants, crustaceans, gastropods, bivalves, and fish. One of the species that we focused in on is the standard mussel, because that species there's a lot of data worldwide and throughout Alaska on concentrations of contaminants in mussels. So we've looked at them, looked at seasonal patterns of contaminants within the mussel tissue, and again have a pretty good indication of a very clean ecosystem. So to summarize the oceanography, we have moderately strong circulation in the bays, driven by the tides, and we have relatively high tidal ranges, not as high as northern Cook Inlet, but quite substantial. We see that local inputs to water chemistry appear to be less important than the regional input from the upper inlet. One thing I didn't mention, the temperature varies from 0 beginning in November up to maybe 15 degrees centigrade in the late summer. For chemistry, levels of trace elements, aliphatic, the straight chain hydrocarbons, polynuclear aromatic hydrocarbons, are very typical of background as represented by data from other Cook Inlet sites. So we have little indication of contamination caused by activities of man in either the water, the sediments, or the biological tissues. So enough of the chemistry lesson. Let's go on to something a little bit more tangible and perhaps recognizable to everybody. And this is the benthos. This is plants and animals that live on the bottom. And we started out working on this in 2004 with participation of those named individuals, including Steve Jewett from UAF, who was one of the divers in 2004. The diver transects are looking to identify important habitats and the species associated with those habitats that are difficult to sample remotely. If you have rocky areas or areas with deep boulders on the bottom, you can't just drag a net through it or drop a grab in it and hope to characterize what it's like. So you have to get down there and take a look. And then we've also done in-fauna, animals living within the sediment, both intertidally and subtidally. In the intertidal, we were building on a background of data that we began collecting in the 1970s. That was, I think, before Pebble started. And we were out there again in 1996 with funding from CIRCAC, the Cook Inlet Regional Citizens Advisory Council. And then beginning in 2004, we began to collect data on additional sites as well as going back to our earlier sites in order to get quantitative data on the beaches so that we could understand any changes that might be occurring. And we wanted to identify important habitats such as kelp beds, eelgrass beds, rookeries, and areas of herring spawning. I might mention, and probably should have in preface, that what I'm going to be talking about is all of the critters there except for birds and mammals. That those organisms are covered under a separate contract with ABR, Alaska Biological Research. So the area sampling history, as I mentioned back in 1970s, under the Outer Continental Shelf Environmental Assessment Program, Dennis Lees and I and others worked on a number of the same places that we have our present sample stations here around Knoll Head, out on Scott Island. We had a station at White Gull Island. And so we have been, and then again in 1996, we went back to several of those same places and did exactly the same kind of sampling. So we have very comparable data from the late 70s and from 1996, and then we began to build on that in 2004. So looking at the nature of the habitats we have, much of the shoreline is very rocky, at least at the upper intertidal, where you have steep mountainsides coming down to the beach. You have a lot of rockfall off of those mountains, and that tends to leave some beaches covered with boulders and jagged angular rocks. We also have some fairly substantial stressors, things like ice beating on the shoreline. We have the rock weathering, so the rock is disintegrating and coming apart. We have quite an exposure to Kamishak Bay swells that are basically coming in from the Gulf of Alaska. So there's a lot of heavy wave action, and then there's a fair amount of local wave action that's generated with very strong local gusty winds that are typical in these fjord-like situations. And the wind waves then also create high turbidity occasionally, where you've got a big broad mudflat and waves breaking on that mudflat kick up a substantial amount of sediment. So as a result of all this, the assemblages, the plants and animals we see in the intertidal zone are not particularly rich or diverse, except for some of the low intertidal areas where we do have quite an abundance of plants and animals present. For the soft bottom shorelines are quite a range of substrate types, including fine sand, silt, mud, if you will, and then other areas where you have coarser gravel. And then some areas are mixed, where you've got the rock in a matrix of sand and gravel. And then there's a few areas of fairly pure sand. Again, we have the same stressors of ice periodically resting on the substrate, freezing onto the substrate, and that strongly disturbs things that can grow there. Our sample locations for the benthic program, a couple of key places, this area of Knoll Head, MPS 1A, that was one of our historic sites that we've resampled multiple times since 2004, as is Scott Island. And while we sort of have shotgunned this whole area because we aren't sure where the port might end up being constructed, so we have a number of stations throughout the area that we've been looking at just so that we have data from wherever a port might be designed in here, and then the other stations can become references against which we can compare future changes. So our field work from 2004 to 2008 has included mapping habitat, especially eelgrass. I've been doing herring spawn surveys, doing rocky intertidal epibiota. Again, these are the critters, plants and animals that live on rocks, and they're often used as an indicator of change. In the soft intertidal in fauna in the sand and mud flats, and the tissue sampling that I mentioned earlier. So looking at the approach we've used, somebody yesterday mentioned stratified random sampling. That is what we have been doing here. We pick an elevation that's typical of a biological zone and lay out a tape measure, pick random numbers along that tape measure, and put down these quadrats and basically count and estimate percent cover for everything there. And this is an approach that we started using in 1970s, used very extensively during the Exxon Valdez oil spill, and have continued here. So one of the things that we've seen is that there is this strong seasonal signal where during the winter you have a lot of these icebergs that are being rocked on the beach by waves, and you can see this piece of rock right here is pretty much denuded of any plants or animals. The little piles of white that are collecting snow are actually the residual rockweed that's still there in the spring. But because of this large interannual variability and the time-intensive nature of the work that we do, we're sort of relying on some data that we gathered in 1978 to describe how that seasonal pattern plays out in these areas. So this is a look at the pattern in 1978 at one of our sites, looking at brown algae, red algae, and encrusting algae over time, April, June, July, and September. At the upper intertidal, in this particular site, it's a very rough, angular rock, and so the ice doesn't really affect things as much as it does in smoother rock areas, like at the middle tidal elevation, where ice can pretty much clean off the entire rock surface, except for maybe the tiny little base of some of these algae, particularly the red algae in the genus Palmaria, which get scraped down and then pick up beginning in April and grow to peaks during the summer and then decline after they release their gametes in the summer. So the nice thing about this is we have this picture and we don't expect that has changed, so we've been trying to focus our current sampling in that midsummer period in June and July, when things are pretty much at their maximum. So looking at the upper intertidal of a typical shoreline, again, here we see a very rough rock surface. Our transect line is laid out. We pick our spots to sample, and we return to those same spots year after year. But basically this area is characterized by a fairly simple assemblage of barnacles, limpets, littorine snails, and red algae. We have a few mussels and patchy rockweed, fucus. And then in some of the more sheltered pockets where water stays during low tide, you have much more life present. You have this is pink rock is an encrusting coralline algae, more red algae, more hermit crabs, and so on. Here's an example of how we get back to the same quadrat each year. This is one quadrat of 10 that we sample at a given elevation. We try to do 5 to 10 quadrats at each elevation. But with the aid of photographs, we've been able to get back to the same place pretty well. Now with the new GPS, we can get back within 6 inches of the same place. But we also mark the rock with either nails or epoxy so that we can be pretty sure that we're right back on the same quadrat. And that helps us, it cuts down on the sampling variability so that we can look more closely at temporal or time series patterns. One of our sites here, this is a knollhead site, middle elevation. The middle elevations are typically dominated by algae. If you go to a place like this, you have to look for a long time to find any animals whatsoever. It's just a very strongly algal-dominated community because of the nature of recruitment to the area and the predator. There are predators here that will eat any animal that tries to become established in this zone. So we end up with a very strongly algal-dominated assemblage. Again, here's a time sequence from the 70s through 2005, and then again in 2008. So an interesting thing that we noticed when we went back in 2004 at this particular elevation on 2 of our stations. In 1978, these stations had both been very strongly dominated by red algae. And in June of 1996, when we went back, there was still very similar assemblage, very strongly dominated by red algae. But then we went back in 2004, lo and behold, there was a co-dominance. The red and the rockweed were very equally abundant. And this was quite a surprise. And what we think occurred was in that early 2000 period, there was a series of fairly low ice years. And so the ice that would normally rip the rockweed off the rock didn't occur, wasn't as effective at ripping that rock off, ripping the rockweed off of the rock. And so rockweed, which is a perennial and it takes 2 or 3 years to get to a mature state, had in fact done that by 2004. By 2005, at Scott Island, that same pattern was evident. So apparently in winter of 2004, Scott Island didn't get the ice effect that had apparently returned to the Knoll Head site, where again we're back to having the red algae dominated. And in fact, since then, in 2008, you see the basic picture looks very much like it did in the 70s and 90s. So turning now to the lower rocky areas, much more diverse assemblages, a lot more different species of algae, a lot of kelp, more diverse animals, including hermit crabs, shrimp, chitons, sea stars. Blennies is a little fish that lives in under the rocks at low intertidal. And the lower intertidal also is important as a location for herring spawning. And some of the areas have pockets of sand and gravel within the rock habitat, and there you get more typical infauna of clams and worms. Diver transects looked at a number of features, including bedrock reefs, boulder flats, and some mixed areas. When we say mixed, we're kind of talking about this situation down at the bottom slide, where you have a lot of cobble-sized material, but it's in a sand-mud matrix. So you have both the epibiota living on the hard structures, and you have infauna living within the sediments themselves. In the intertidal areas, we do typically sample 2 elevations, trying to get the lowest elevation down near mean lower low water. We photograph the quadrats, and we do density and cover, and then we take a core with this highly technical device here in the lower picture, and it's actually a sawed-off clam gun. But we take that material and sieve it on a fine mesh sieve and sort out the infauna, the animals living within the sediments. And then the mega infauna is a larger full quarter-meter square quadrat that we basically shovel through a coarser screen, and we use that to get quantitative information on the larger clams and worms that may be present. So the soft shorelines, as I indicated, are very extensive. This is looking up Inniskin Bay. These mud flats at low tide can be half a mile or wider, and extend for multiple miles up the bays. A typical quadrat in the infauna are looking at the soft intertidal. You can see little tiny sprigs of eelgrass growing here. And eelgrass, I'll talk about in a bit, but like the rocky intertidal assemblages, the soft intertidal is also subjected to the same ice scour, freezing and lifting of the surface materials. So we end up with very few plants on the sand and gravel substrates, because those are areas that are getting wave washed on a daily basis. So nothing, not too much grows in those areas. But in the mixed intertidal and in the mud flat or the sand flats, we have a pretty interesting assemblage. Large areas of eelgrass, but the dominants in the infauna are bivalves, clams, marine worms, and also hermit crabs will be present during high tides. Typical creatures in the infauna for the intertidal, we have highly variable substrates in our sample regime, but within each station the habitat type is relatively uniform. Again, have to keep mentioning the wave stress and the ice stress. So as a result, the intertidal infauna is much less rich and diverse than subtidal infauna. But because of that low flat gradient, there are relatively few predators there, and that helps allow the larger animals like the clams to survive, because they don't have sea stars that are preying on them. In the dominants, in terms of biomass, several, mostly clams, echurus, and other polychaete or marine worms. Subtidal infauna is fairly similar to the intertidal in terms of the major taxa present, but it is much more rich and more diverse, more species present than in the intertidal. and it does have the predators, sea stars, that will alter the community by eating selectively on certain species. Okay, for summarizing the infauna, worms, mostly the genus Nephtes, are very abundant, and mollusks, mostly Macoma, are the dominant two groups within the infauna. Crustaceans are usually only present in the form of amphipods. We haven't found a lot of the other crustacean groups that are typical of infauna in other areas. So because of the geographic spread of our stations, there's quite a difference between each site. So it's not easy to say that one site fits all. So there's several different types of soft bottom habitats, and they're each come up with a different characteristic. A couple of notes on the habitats of special interest. Kelp is interesting in this area in that we have very dense kelp beds of the genus Saccharina and Alaria, but we don't have the canopy beds of bull kelp or Macrocystis that you find on the other side of Cook Inlet in Kachemak Bay. And that, again, not quite sure why that is. I expect somebody probably knows. But eelgrass, here's one of our more robust eelgrass patches. This is a place we call Blackie Beach, and this eelgrass bed is very similar in this photograph to what it looked like in 1978 and 96. But when we went back in 2004, there wasn't any there. It was gone. But then in 2005, it came back. So eelgrass, again, is influenced by these short-term events of perhaps bad ice or high winds from the wrong direction, which can remove the entire beds, but they do seem to regenerate. Typically, the eelgrass occurs in very small patches on the order of 5 feet or 10 feet in diameter. This is Blackie Beach in the photograph. But we have a very extensive coverage. I don't know, can people see the bottom line there? Maybe we should move those chairs. But anyway, about 450 acres within the two bays of eelgrass, but that eelgrass is not continuous. It's again, in relatively discrete and small patches, perhaps covering as much as 10% of the bottom area within that 450 acres. So the next habitat of special interest is herring spawn areas. Fish and Game has been doing spawning surveys in this area from 78 through 94 using both aerial aircraft surveillance to look for schools of herring and evidence of spawn in the water. They also do ship-based spawning where they do purse seining and look at the condition of herring that they catch. But after the early 90s, when there was a commercial harvest, herring basically declined in this area to the point where there has been no harvest since the early 90s and very little spawning activity documented through 2004. In 2006, we looked perhaps too early in the season and didn't see any spawning. But beginning in 2008 and sneaking in a little peek of the future, 2010 to 2012, we did find substantial herring spawn in the study area. This is a kind of a map that shows the tracks that we've followed. We do these surveys boat-based with a couple of observers in the boat at extreme low tides, hop out and walk along the beaches and look for the spawn. So the purple here are areas where we didn't find any spawn in this particular survey. This was from 2010. Interesting note, in none of our surveys have we found significant spawning along this piece of shoreline on the peninsula between the two bays, nor have we found any in Iliamna Bay. So the spawning appears to be concentrated from an area just beneath Knoll Head, which is this peak here, and then up into Inniskin Bay on the right side of the picture. The spawn out here is on red algae and rockweed. It seems herring in this area seem to spawn preferentially on rockweed, which is interesting. Then up into Inniskin Bay, this is an area of patchy eelgrass, and the herring in that area are spawning on the eelgrass. And then over on the other side of Inniskin Bay, Fossil Reef and Blackie Beach are again areas of consistent spawning. We have in other years also found spawning out around the Mushroom Islet here and Scott Island. So it appears that the herring may have recovered a bit from their depression during the 90s and early 2000s, and that there may be a significant population in the study area now. So if we're looking at the littoral, which is the near shore summary, we have diverse habitats from rock to basically mud flats. We have a high stress environment. We have low to moderate primary productivity and low to moderate biological diversity overall. We do see a strong coupling to demersal resources, which means that the shallow water habitats do provide food resources and energy resources to other resources, to deeper water resources, and I'll talk a little bit more about that in the section on fish. So the probable importance of these areas to the adjacent Lower Cook Inlet ecosystem. These shallow embayments, like shallow embayments everywhere, provide a substantial amount of organic matter in the form of algae, algal growth that is fixing the energy of the sun, if you will, and exporting it through tidal action out to other marine areas. This area is important spawning and rearing area for Pacific herring. We've also see that it's an important nursery area for salmon, juvenile salmon, other forage fish other than herring, including smelt and sand lance, Pacific halibut, and crab, including tanner crab, are very common in there. So they're all feeding on something that's related to that littoral ecosystem. So, on to fish and demersal invertebrates. These are both fish and invertebrates that move in and out with the tide and are feeding and, in many cases, growing in the area. Our goals here are similar to our other goals, building on historic data with comparable sampling approaches, and we are seeking to gain an understanding of how fish and invertebrates use these areas seasonally. And for previous work in the fish realm, as part of the same OCSEP, or Outer Continental Shelf Environmental Assessment Program, work, Jim Blackburn et al. Did a detailed fish survey within Lower Cook Inlet, and the unfortunate thing is, well, he gathered data from both Kachemak and Kamishak bays. In the written report, it's difficult to tease out the data to identify and look at the data that are co-located with the sampling that we've done. But we do get some insight from looking at that work. And I mentioned the fish and game herring spawn surveys, which I'm not sure that they have funding to continue. That could be of concern. So our objectives: describe these fish and invertebrate assemblages, clarify their seasonal patterns. We did seek to sample throughout the ice-free time frame, look at feeding relationships, and I'll talk a bit about that, and then also get our tissue samples for the background chemistry. So our field work, one of the primary tools we used is 37-meter beach seine shown here. This is a net that Cy actually kind of developed and standardized as a prime tool for doing this kind of field work in Puget Sound, and we have been using it for all of our work up here. So the beach seine, we also use an otter trawl, which is like a commercial trawl or bottom trawl, but much smaller. And then we use gill nets and what the thing called a trammel net, which is sort of like a gill net on steroids that has separate panels to entangle larger fish. And then we also are doing the water chemistry that I talked about and getting the tissue samples. And looking at when we sampled, we started out in August of '04 basically doing a reconnaissance of the area. And then in '05 we sampled from May through August, and we began to get feedback from the interagency meetings, the annual meetings that were held, that we needed to look beyond May through August. So we did April and September in '06, then September and October in '07, and then finally in '08 we actually got out there every month from March through November, and that led to some interesting sampling. For the fish and invertebrate, demersal invertebrate work is pretty well scattered throughout these two bays. The yellow dots here are 120-foot beach seine sample sites. Don't remember exactly how many, but quite a few there. Then we also use a smaller 30-foot beach seine in a couple of little lagoons that we've been sampling. And then the green dots show where we've done our bottom trawls, and the orange are the gill nets and trammel net sites. The reason we use the gill nets and trammel nets is to sample areas that are basically rocky, where you can't actively pull a net through the area because you get hung up on rocks. So they're what's called passive gear, and you set them out there and capture fish that are moving around on this rocky or bouldery substrate and get into the nets. So the shoreline use by fish, as indicated by our beach seine, we get salmon, both juveniles and adults, that use this area for our migration feeding. Osmoregulation, because it's low salinity when they're coming out of their spawning streams. Freshwater, they need an area where they can slowly adjust to living in salt water, which is quite a different challenge physiologically. And then we also have very broad, low gradient areas that can be used as refuge from predators. Forage fish in this area, a very heavy use by herring that I'll talk more about. Surf and longfin smelt are quite common in the area, and Pacific sand lance as another forage fish that is certainly important in local food webs. And then there are the flatfish, the flounders, halibut, skates that are also abundant in the area. And then a group called codids or sculpins or bullheads that are not particularly commercially useful, but are an important part of this community. If we look at the seasonal catch pattern, this graph shows our catch per unit effort, and that's basically catch per set with the 120-foot beach seine or the 37-meter beach seine. Over time, that's the pink line here, so beginning in March, we have relatively few sets in March, but we did catch a substantial number of herring, age 1 herring, in one of those March sets that makes that catch per unit effort fairly high value. Peak in catch per unit effort is clearly in June, and that is driven by, again, the age 1 herring, which can be extremely abundant along these shorelines in June. Generally, the catch declines after September as weather gets worse, the water gets colder, and a lot of these fish move offshore or into deeper waters. And then the blue line here just shows the diversity of species that we see in the beach seines through that annual cycle. Looking now just at juvenile salmon, here we have 3 species of juvenile salmon plotted in Iliamna Bay, in Iliamna Bay primarily driven by chum salmon, which spawn in several of the local streams, followed by pink salmon. They both peak in May in this particular bay, and we get a few sockeye coming in from other areas in June and July. Then in Inniskin Bay, we have quite a different looking picture, and in part this is because in Iliamna Bay we're sampling well up into the head of the bay where these fish are coming right out of their spawning streams. But in Inniskin Bay, most of our sampling is in the outer half of the bay. So what we're seeing there are fish that have been out of their streams for a while. And for example, the pink salmon are quite abundant at that location in June and July, whereas they're pretty much declining in the inner stations of Iliamna Bay by then. But we do see quite a substantial growth in the pink salmon. By July, they're fairly large fish. Turning now to herring, this is the annual cycle. Again, catch per unit effort, catch per set, and shows what I was mentioning. In March, we had a fairly high catch of juvenile herring, and those were age 1 fish. Again in June, very, very high catches of age 1 fish. Most of what we get are age 1, although by late in the summer we're starting to pick up young of the year, very small herring. And this is what's called the length frequency histogram that shows the average sizes of these fish that we catch. And through the year, and this I think is all from, this is all years combined. So as you can see, we had only three beach seine sets that we've been able to do in March, but we did get that one strong catch of age 1 herring. And that group of fish, that cohort of fish, was evident in later sampling into August. And then by July, we start to pick up very small fish. These are fish that are probably on the order of an inch long. And they're from recent local spawning. And you can see as the average or the median size of those cohorts increases, and what we call apparent growth of the fish through that time frame. Then looking at the same type of data in the otter trawls, the bottom trawls, we see an interesting movement pattern wherein during the summer, early summer, May, June, July, we basically catch no herring in the trawl. At that time, they seem to be pretty much focused on the beaches where we pick them up in great abundance in the beach seine. But by August, we start to pick up a young of the year cohort in the trawls. And even into November, March, April, we still are getting this cohort in the trawls. So what they seem to be doing is after the very productive period of midsummer, they begin to move out into deeper water and frequent benthic habitats or near bottom habitats, not to the exclusion of the shoreline, but basically they're really spreading out and using all of those habitats. They were still present in the shoreline areas in March and November, so it's unclear. Well, it appears that they really are using the full range of depth that's available in that time throughout the winter. So, looking at the trawls, other species that we catch in the trawls, flatfish, yellowfin sole is our third most abundant, starry flounder, Pacific halibut. In the gadids or cod family, walleye pollock is the fifth most abundant in our trawls. The most abundant is a critter called the snake prickleback, which is not on anybody's high domestic use list, but nonetheless is quite abundant. Herring is the second most abundant species we get in the trawls because they're basically present, except for that summer period, throughout most of the year. And then a number of other groups that are also taken. Looking again at the timing of these different fish species in the bottom trawls here, the red line is the herring. Again, we can see the peak in March and then again in September, but a general absence of herring in those bottom trawls throughout the summer. Snake prickleback, the most abundant species in the trawls, essentially has two peaks, one in the spring, and those are mostly larger fish, and then by August we start to get young of the year snake pricklebacks in great abundance in those trawls. And then the other species that are graphed here don't show particularly strong seasonal patterns, except perhaps for walleye pollock is more of a winter, early spring, late fall component in that catch. In comparing our beach seine information with Jim Blackburn's data from 1978, we put this together just to kind of look at the ranking of fish abundance in Jim's data from the 70s compared to ours. Generally not too far off. A couple of anomalies are evident that he, for example, got a lot more surf smelt than we have. We do get a substantial number, or excuse me, we get more than he did, and we're not quite sure why that is. We've actually been trying to figure out if surf smelt may spawn in the study area, but we have not yet found actual evidence of, or direct evidence of spawning, that is, eggs, in the beach. Let's see what else. Herring were much more abundant in our studies than in his. And one species that we haven't caught at all is capelin, and they were fairly common in Blackburn's beach seines too. So anyway, we do have some nice opportunities to compare sort of long-term trends, but we're hampered a little bit by the fact that as you see there, the rank from Blackburn's work includes both Kachemak and Kamishak bays, so there's not a direct comparison there. So looking at invertebrates that are present in the beach seines, we have a lot of small shrimp-like things. Mysids are extremely abundant in many cases. Crangon is the little shrimp shown in the lower right-hand photograph. Gammarid amphipods and other small crustacean are quite common. And occasionally we get very high abundances of a couple of what we might call jellyfish that are actually pretty different animals. In the trawls, again, we get crangon, the sand shrimp, probably one of our most abundant invertebrates there, and a variety of other plants and animals as listed there. The macro inverts, there's some interesting patterns in that there we seem to have higher densities of macro invertebrates in our trawls in the spring and fall than we do in the summer. We don't know if that's driven by heavy predation during the summer or if it's a matter of these animals moving into different parts of the Cook Inlet environment. But what it amounts to is that it does show that in the winter there's a very substantial invertebrate population, and that's important because a number of species overwinter in this area. Things like stellars, eiders, and sea otters overwinter in this area, and this suggests that there is some food there for them to eat, and that's probably part of why it's an important overwintering for those animals. I'm looking at the overall catch by station of crangon, the sand shrimp, and pandalis, which is the shrimp that we typically eat. The big thing that stands out are there's two of these stations, MTR4 and MTR3, are both trawl stations up in the innermost parts of the bays. And what we see from this pattern is that in those areas we don't get any of the pandalid shrimp, we only get the crangon. And again, this is of interest because those inner parts of those bays are the important areas for bird and marine mammal overwintering. And let's see, this just looks at the rank of the four most abundant animals. These are all crustaceans or various sorts of shrimp-like things. But basically these four genera, or these four taxa, always rank either 1, 2, 3, or 4 in those catches. And this photograph in the lower right is literally a bucket of shrimp that we got in one trawl one time in August. So that's why that probably attributes to the fact that pandalis was the most abundant and seems to be the most abundant through that summer period fairly consistently. Another species we get, interestingly, only in the fall is the tanner crab, commercially important species, and we find a fair abundance of these relatively small tanner crab in there, particularly when we've sampled in September, October, and November, and those 3 different months shown in that bar graph. Another interesting aspect of the tanner crab is it's one of the most abundant species in stomachs of halibut in this area. So that may be why we don't catch them very much in the summer, because summer is when halibut move in, and they may be pretty effective at eating these little guys. So looking now, I mentioned coupling of the food web between fish and benthos. And so this is a pie chart that shows the dietary habits of starry flounder in the study area, and quite an interesting dominance of bivalves and polychaetes in the diet of those starry flounder. But if you look at a very similar fish, a yellowfin sole, it's completely reversed, where we have polychaetes being extremely abundant in their diets and bivalves being much less so. Pink salmon, another example of who's eating what. Pink salmon seem to take about half of their prey as terrestrial insects and almost half as other arthropods, copepods, planktonic or epibenthic copepods that are very small crustaceans. Juvenile chum salmon, quite a different picture from the pink salmon, but they seem to have taken an abundance of copepods, but also a substantial proportion of shrimp, these very small crangonid or pandalid shrimp. So, for summary for the nearshore fish, we have diverse habitats. Again, we have the ranging from rock to very soft sand and silt habitats. It's a moderate stress environment even for fish, a lot of wave action, particularly in the spring when you have a lot of very small fish. If they're right along the shoreline, they may get pounded by the breakers from the large swells coming in. We've shown highly seasonal use, a lot of variation from the winter period, the shoulder periods of late fall, early spring into the summer, and highly variable fish densities through that period. Again, looking at the importance to the Kamishak and Cook Inlet ecosystem, herring spawning is certainly a big one, and we have demonstrated this area as a nursery for salmon, forage fish, flatfish, and crab. We have also seasonal feeding by a spiny dogfish, big skate, and Pacific cod that we don't collect very well with our sample gears, other than angling, hook and line sampling. So, I guess that's the end of it, and lest you think the weather's always like that, we'll leave that one up.
All right. Well, thank you, John. I'd particularly like to thank you for giving that presentation in English instead of jargon. That's very appreciated. We're scheduled for a 15-minute break now. Let's shoot for about 10:15, and then we'll come back and do questions and comments from the panelists and then the public.